Say when you're doing a lot of exercises or not eating enough carbohydrates, your body is run out of glucose, the primary fuel source, to provide energy. Your body switches to fats, the secondary fuel source, to generate energy for your cells. Triglycerides are the main storage form of fats within our body. The mobilization of fats starts with the breaking down of triglycerides into glycerol and fatty acids. The fatty acids are taken to different tissue cells. Many cells can use fatty acids for energy, but the three big ones are your heart muscles, skeletal muscles, and the liver. So let's say in a healthy person, the free fatty acid is brought into a general cell. Here you see there is a cell membrane, there is a mitochondria, and the mitochondria has two membranes, the outer and inner membranes. Outside the outer membrane is cytosol, inside the inner membrane is matrix. There are some enzymes and translocases located on those membranes. Let's say there is a 10-carbon free fatty acid that we want to bring into our cell. We know that it is a medium-chain fatty acid because it has in between 6 to 12 carbon atoms. Now we bring it into the cell. The carbon attaching to the double bond O is number 1 carbon. The number 2 carbon is right next to the number 1 carbon, and it is also called the alpha carbon. And the number 3 carbon is called the beta carbon. The first thing we have to do is to prevent this free fatty acid from leaving the cell. To do this, we use a special enzyme, the fatty acyl-CoA synthetase, to add a CoA on it. This process uses energy. As a result, a fatty acyl-CoA is formed. So now we have activated the fatty acid and can keep it inside the cell. But we have another problem. We cannot transport it into the mitochondria because of the CoA. This purple enzyme produces a special molecule called carnitine. The carnitine combines with the fatty acyl-CoA while getting rid of the CoA. As a result, we form a structure called fatty acyl-carnitine, which is transported through this translocase into the matrix. But we have another problem again. This fatty acyl-carnitine can easily go back out because this is a bilateral translocase. To prevent this from happening, we have to rip that carnitine off and then add another CoA back onto it. This purple enzyme here takes this CoA and add that on this fatty acyl-carnitine. At the same time, it rips off the carnitine. As a result, we have a fatty acyl-CoA inside the matrix. Then you may ask, what are those purple enzymes? On the cytosolic side of the mitochondria, we have this enzyme called the carnitine acyltransferase type 1. It adds carnitine to the fatty acyl-CoA so it can be transported into the mitochondria. Once inside the mitochondria, to prevent it from getting back out, it has to be acted by this enzyme which pushes the carnitine off while adding a CoA on. This another enzyme is called the carnitine acyltransferase type 2. Now we have a fatty acyl-CoA. We can undergo the next step. But can you recall what were the two steps we have just performed? First, we get the free fatty acid inside the cell. We convert it into the fatty acyl-CoA through a step called activation. The second step, we transport the fatty acyl-CoA into the mitochondria so we can undergo the next step. This next step is the focus of today. It is the beta oxidation. In the first step of this reaction, we are going to remove a hydrogen from both the alpha and beta carbon. Who's gonna do this? FAD. And it turns into FADH2. What enzyme is catalyzing this? Since the enzyme is taking up hydrogen, it must be a dehydrogenase. And because the substrate molecule is an acyl-CoA, the name of this enzyme is called acyl-CoA dehydrogenase. The result is the formation of a double bond between the alpha and beta carbon, and this resulting product is called enoyl-CoA. Now in the second step, we will add a water molecule to the double bond of the molecule. In other words, we need an enzyme to hydrate it. This enzyme is called enoyl-CoA hydratase. We add an OH group to the beta carbon and a hydrogen to the alpha carbon. Since the hydroxy group is added to the beta carbon, the product is called beta-hydroxy-acyl-CoA. Then in the third step of the reaction, we use an enzyme which helps turn NAD positive to NADH. This enzyme takes two hydrogen on the beta carbon, so there is a formation of a double bond between the beta carbon and the oxygen. As a result, a ketone group is formed on the beta carbon, and this molecule is called beta-keto-acyl-CoA. The enzyme catalyzing this is called beta-keto-acyl-CoA dehydrogenase. In the fourth and final step, we add a very special enzyme called the thiolase. It does two things. First, it cleaves the bond between the alpha and beta carbon, and the second, it adds a CoA to the beta carbon after that bond is broken. What are the two products formed? As mentioned, thiolase adds a CoA to the beta carbon, and this forms a new fatty acyl CoA. Where is this fatty acyl CoA going to? It goes back over here and get recycled, and it will go through another round of beta oxidation. The question that you might ask in the beginning is, why is this reaction called beta oxidation? The answer is, we are actually breaking down the bond between the alpha and beta carbon to release a fatty acyl CoA, which is 2 carbons shorter. And in this case, since the original fatty acid has 10 carbon, now the regenerated fatty acyl CoA has 8 carbon. So what is the other product in the reaction? A hydrogen attaches to the alpha carbon, and guess what? An acetyl-CoA is formed. It is a 2-carbon molecule. So what happens to this acetyl-CoA? We know that it will enter the Krebs cycle and release ATP, NADH, and FADH2. And NADH and FADH2 will go to the ETC and leads to oxidative phosphorylation. The overall result is ATP formation. And do you still remember one FADH2 and one NADH is produced in step 2 and 3? They are fed into the electron transport chain as well to produce more ATP. The regenerated fatty acyl CoA also repeat the beta oxidation again to yield more ATP. So when glucose is depleted, healthy people can utilize fat for energy production. However, what if this acyl-CoA dehydrogenase is deficient? The whole beta oxidation will be impaired. Patients with medium-chain acyl-CoA dehydrogenase deficiency cannot convert the medium-chain fatty acid into energy. And what problem will arise from that? We will find that out in our further PowerPoint presentation.